I'm Karine Labadie from the Genoscope, and Laurie Bertrand here from the Genoscope also. So, sorry. I will try. Just few slides from the Genoscope to present the institute and then the Cryogrinding, but just I will spoil some slides, but we use the biomeasure as Irene showed you from the Sanger, but we also use other things. So I will present you that. For the Genoscope, sorry, for my voice. For the Genoscope overview, we are the French National Sequencing Center, which is led by Patrick Banker. And we are part of the CEA Institute since 2007. We provide throughput sequence to the academic community and carry out in-house genomic project. And we are focusing on biodiversity, with the de novo sequencing and metagenomic projects. So few slides from the metagenomic project, BGE and ERGA, but also all the biodiversity of Europe. The Atlas project, which focuses on the marine biodiversity in the France, coastal France, the EEZ, the French EEZ. And for the metagenomic project, it's all the projects with Tara Ocean project, which starts in 2009. So for the expertise, we have expertise from sampling, because we are on the field in the Atlas project and collect all the biological tissues. The wet lab, for the wet lab part, the DNA and RNA extraction, IC preparations, and NGS sequencing, short read with Illumina NGI, and the long read sequencing with ONT and the PacBio. Yeah, just a picture of the wet lab team with Laurie here. And for the dry lab, for the genome assembly and genome prediction, gene prediction, and the picture of the team of the dry lab. And just here is Caroline Belzer, who perform all the QC validation and the bioinformatic validation from the IC libraries. So just a picture of what we did from the beginning. So as Irene said, we manage a lot of different samples from plants, blood, mammalian and non-mammalian samples. In blue, you can see the ARIMA technology. We start many years ago with the 2 enzyme kits with plant tissue and animal tissue, low input and standard input. And after, I think 2 years after or 1 year after, I don't know, it was the ARIMA IC with 4 enzymes. And we also manage the IC protocol from Dovetail. So we start with the IC from Dovetail, then the OmniC kits from Dovetail. Also for plants, blood, mammalian and non-mammalian. So just to begin with the tissue weighting and grinding. So as I told you, we did a lot of ARIMA low input protocol and OmniC protocols. So for the ARIMA low input, we use mainly the biomeasure, like Irene. So I won't speak about the biomeasure. Just for bigger samples, more than 50 milligrams, I think, 50 milligrams to 2,000 or more. And I think since 2 grams of leaves for plants. We used a mortar and pestle. So just here is a picture of the organization of the benchtop when we start the manipulations. So we have the dewar containing the biological samples that we put in liquid nitrogen, sorry. We have here the mortar and pestle that are pre-cooled. So we put the day before the mortar at -80, and then the day of the manipulation, we just put in an ice bucket. Just another ice bucket with the pre-cooled tube, and then a precision weighting scale at 10 milligrams. Here's a picture of the cryogrinding steps. So first, we cool the mortar at the top. We cool the mortar with liquid nitrogen. Just use a tar. What is the name of the big spoon in English? Thank you. I don't know. And then we just place here the sample. You can see here the sample. In the mortar, we crush the sample with a pestle, cryogrind the powder. So here you can see before it's cleansed. Here it was the cnidarian sample. No, isn't it? Corallite. I think it was a cnidarian sample, but here it's a leaf sample before and after the grinding. And then we recover the powder in a pre-cooled tube using the blue spatula. And then we weight the tube using a tar on the precision weighting scale. But we also use sometimes other options for the breeding. We also use 2 times or 3 times the fast prep. We tested on the amphipods and the zooplanktonic organism, because in the frame of TARA project, we have a pool of marine organisms spotted on the filter. And then we take all the organisms and then we put them inside the tube, and we perform the meta-IC with that. So we use the fast prep in the RMIC animal tissue low input after the crosslink, and you use the VK05 bits plus bits with it's just 0.5 microns and 0.1 microns. Just a pool of that bits. It's small, very small bits. We also use the Cryogrinder set like that, but we are not very satisfied for this kind of set. Because we can see in real time how effective the grinding is, and it's difficult to recover the powder after the grinding. And it's very weakness, so we never use it. We have it, but we never use it. And we test the cryoprep Covaris, and we feel that it doesn't grind into fine powders. So it's the results for plants in the cryoprep. So I don't know if everybody know what is the cryoprep, but the slide was sent to the, okay. So you can see on the site, it's just this machine, and you have 2 hammer, and they crush the sample inside. You put the sample in the bag, and then you recover the powder into the tubes. But for us, for plants, for example, we are not really satisfied because it's not fine powder like the cryogrinding and mortar and pestle. Also, it's difficult to recover crushed sample because they stick on the surface of both the bag and the tube. So if you want to crush again inside, you can recover the powder here to put inside the bags and to redo the manipulation. So we are not really satisfied. And for corals, here they are not in powder, and also it tears the bag when we crush the corals. So I don't know if you have the same experience as exactly, and you continue to use it.
We use the fast prep.
The fast prep for corals.
Corals, plants.
Plants also. Okay. So for us, we just we test it with just the vortex at the beginning. Just we put the samples with beads and with vortex because I manage also the DNA extraction, high molecular weight DNA extraction. And when we use the fast prep for high molecular weight DNA extraction, if you use strong protocols, we shear the DNA. So we prefer to use just the vortex with a sample in beads. But for the HiC, we tested the vortex and we never joined the amphipods. So Laurie used the program that we use for the pool of, and it's a strong program. I don't remember the cycle, but we can tell you the exact protocol, but it's a strong one. But we analyzed the library and we were pretty satisfied. Yes. Laurie managed 2 corals in the past, but she was under the binocular loop, and she picked each polyps. They were in ethanol, and yes, a lot of polyps from the coral, but it worked well. Works well but you spend you spend a lot of time under the binocular loop.
I suppose when the sample is very precious, you can.
Yes,
that.
It was very precious. So we spent a lot of, and we were very happy because surprisingly, we test the ARIMA and the Omni-C protocol, both, in flash freeze samples and ethanol. And we obtained better results with ethanol samples than the flash freeze samples. It was easier because she picked up each polyps and yes. The coral was preserving ethanol and she picked up each polyps, and then we wash to remove the ethanol, and then we proceed with the process. So, and we have pretty good results. Just some examples of the samples that were difficult to green. The elastic samples with mucus, like the snail, it was a pity. The coral sample with a hard exoskeleton. Gelatinous samples with like Medusa, jellyfish, because it's when you have just the mesoglea, the sample, it's just horrible. And here is an example of kitten, but sample with cuticular matrix are sometimes difficult to grade. Just some tips if you participate to the sampling, just sometimes prefer certain organs or soft tissue is difficult if the animals are dissected. Like in the snail, the collaborators gave us also the kidney of the snail, but you have to know better the snail. I'm not the good person to do that, but they give us the kidney, so it was easier to use the kidney for the HiC protocol. And when you sample when you collect the samples, just pre-cut the tissue into small pieces during the sampling because it's easier to grind where they are small. And
When you have a jellyfish,
Yeah.
what organ would you like to have? Optimal?
The arms. We obtain better result with the oral arms and the what was the other one? The mouse? No. The nervous. Yes, yes. Right. Exact. But we managed the best library was with the arm. The arm. Tentacles, yes. And for the IC preparation, sometime after the grinding, you obtain small pieces of material, and we use strainers and filters to remove all the unwanted parts. And it's not sometimes I don't mention the. No, it's another question. Sorry. And that's all for me.